It's a big honor to be here today. Big thanks to Paula and Danielle for putting this together and making this real. Thank you very much. And Myra, thank you very much for your testimony earlier today. Very touching, and it gives a profound meaning to what we do as scientists. It's hard to be the last one, especially when I'm in between you and the lunch. So I'll try to be brief. My talk will be about therapies in Chagas disease. My name is Jair Siqueira Neto. I'm an associate professor here at UC San Diego at the Skaggs School of Pharmacy and Pharmaceutical Sciences. Just to set the ground, I know you're now experts in this, but in Chagas disease, right after being infected, you go through an acute phase, and then you can have some symptoms or not. Most likely you're going to go after in a long stage without showing any symptoms that we call the chronic stage. And later on, about 30% of the individuals will develop some sort of chronic manifestations that could be digestive or cardiac. So when we're talking about therapies for Chagas disease, you can think about the therapies when you reach that later stage with manifestations like cardiac or digestive, in which case you would use treatments to control arrhythmia, the myocarditis, heart failure. So this is not what I'm going to talk here today. My talk will be about therapies targeting the parasite. So really talking about antiparasitic drugs. You know now very well that there are only two drugs that are available for the treatment. These are very old drugs. This started using these in the 70s, and they're still the very only two that we use to these days. I put here the chemical structure, sorry for that, but I just want to call attention for a specific group that is present in both molecules, these nitro heterocyclic or nitro aromatic groups. This is actually the part of the molecule that is responsible for the activity. So what happens is that when these molecules get into the parasites have an enzyme that a group of enzymes called reductases that will activate these molecules. And what happens is that this nitrogen will become very reactive inside the parasite. And they'll react against proteins, enzymes, DNA. So basically cause a big mess in the parasite, and that what leads to death of the parasite. It turns out that our cells also have reductases, and we can also activate these compounds in our cells. And that's why we see all the adverse effects that were mentioned in many talks here. And that's one of the big problems with these two drugs. About 30% of the people can't even handle the entire course of treatment, which is long. So I just want to make it very clear that we do need better, safer drugs for Chagas disease. All right, so how we do that? This is the pipeline for drug discovery and development. I think this became very popular during the COVID time. So a lot of people know already about this. This is not specific for Chagas, so this process will be the same for any disease if you want to discover or develop a therapy, how it works. So it starts by identifying a target, which would be, could be a protein or a receptor, something that is important for that disease in particular. Once you find that, you have to test number of small molecules or compounds that could be active to modulate that biological activity that you know is important for the disease. So that stage we call screening. So you test as many molecules as you want to try to find the ones that are active. And we call these hit molecules. So after you identify these hits that have some activity against your target, there is a phase where you get chemists or medicinal chemists, and they do modifications in the molecule to try to make it even better. So that's the lead optimization phase. After that, you have to show that it's safe, not only in cells that you can handle in laboratory, but also in real organisms, let's say in animal model, because right now we can't we can't model an animal with all its complexity in computer models. So it's still important to test things to make sure it's going to be safe in an organism. And if you have the animal model for the specific disease, you also want to show that your molecule is able to get the expected biological outcome that you want. So all that goes before you even start clinical tests in human. So once you get all that information, you have something that you prove is safe, it's effective in animal models, then you can start testing human to see how it goes. So in the very first phase, we call phase 1 clinical trial, it's usually done not with patients, but with volunteers. And you just want to make sure, it's a very small group of people, you want to make sure that that compound will not cause any harm. So you have to do a lot of studies to predict that, but you still have to go through that phase 1. So you want to assess it's a safe compound. You usually do a dose escalation to see how much you can treat without seeing any adverse effect. If that goes on, okay, you go to phase 2, and that's to test efficacy in a group of patients. So again, you're still assessing if the drug is safe, but now you want to make sure it's also effective to what you're claiming it is. So you start phase 2 with small groups of patients, and then if you get positive results, you move to a third phase, phase 3. It's very similar to phase 2 with the big difference that you're now expanding your population which you're testing, and you try to do this in multiple centers. If everything goes fine, you show all the data to regulatory agents, and they'll say, okay, this is approved, so you can start commercializing, and then that goes to phase 4. So if you're interested in learning all the different trials that goes in human, you can go to this website, clinicaltrials.gov. And if you want to look for Chagas, for example, here's the page. You can just put Chagas as a condition or disease, and they'll list all clinical trials that are currently happen and have already happened against Chagas. This is in the US, the FDA. So even trials that happen elsewhere might also be registered in this database. So you see here 54 trials. It seems a lot, but you're going to see that many of these trials are still with benzinidazole and nifurtimox. So things that trying to reduce the length of treatment or playing with the dose concentration to try to reduce cytotoxicity or adverse effects. This would all be included here. And let's see, if we narrow down to only new chemical molecules, so new drugs, what is there in the pipeline? Well, nothing much. And the two ones that I want to call attention to that were already mentioned, one is posaconazole, that is very similar to the compound that Colin mentioned, the E1224. So you see this molecule doesn't have that nitroheterocyclic group. It's actually a drug used to treat fungal diseases. It's very safe, posaconazole, and with all the studies in cells and in vitro, it has a very good efficacy against Trypanosoma cruzi. So about 10 years ago, there was a big hope that this could be the future effective and safe drug against Chagas. Well, unfortunately, in the clinical trial, once they enroll all the patients, they treated them. They had 2 doses of posaconazole, and they had benzinidazole as a control. Right after treatment, everybody was still negative. This was tested by PCR, so it seems like the treatment was very effective. But when they follow these after a year, what happened is that the efficacy of these posaconazole, both groups at 100 and 400 milligrams, decreased a lot. So about 80% of the patients became PCR positive again, which means the treatment was not very effective in the long term. The parasites were still there. So it was a big disappointment. In the same a trial with a very similar molecule mentioned by Colin happened at the same time from a different group. So now we know that molecules that are similar to these that target the same thing is a protein in the parasite responsible for synthesis of ergosterol. That's part of the membrane. They're no longer of interest. So every time you come up with a new compound, you have to test if it works on the same pathway, and if it is, it's deprioritized. The other compound that I want to mention that was in the clinical stage of development is fexinidazole. So about 10 years ago, they did the first phase 2 trial, and they saw some issues with safety, so they stopped the trial. And this is very common. You saw the same thing for COVID sometimes. During the trial, if you see something that calls attention, you stop, you analyze, and then you decide to go or not. And in this case, they decided to extend it for another 12 months. And they got some interesting conclusions that they still see some sort of adverse events. You see here the nitroheterocyclic group in this molecule, but it seems like if you shorten up the treatment and reduce the dose, it would still have the good antiparasitic activity, so it might be safe. So they decided to do another trial, and this was completed, I believe, the end of last year, and we're expecting the results to be published soon, so in the coming weeks or months. So that's the expectation, and these are really the only two new chemical entities that were in the pipeline for T-Cruzy. So now I'm going to talk a little bit about my research, what I do. So my lab is focused on this very early stage. The goal of my lab is to feed this pipeline with as much as new candidates as we can, and we can go all the way here to a preclinical candidate. And from that moment on, institutions like the DNDi would partner and run all the clinical stage. So how do we start? How we identify these targets? Well, it's all about trying to find the weak spots from the parasite, really. So there are two ways you can do that. You can actually try to, based on basic science and a lot of fundamental biology of the parasite, try to identify things that are essential for the parasite, and they are druggable, meaning you can use a small molecule to target that and get a good outcome in terms of treatment. The other way to do it is what we call target agnostic approach. And in this case, you can infect a cell in a lab with a parasite and try different compounds to try to kill only the parasite, not doing any harm to the whole cell. Even if you don't know the target, you could still get some molecules that are potentially good drug candidates. So I'm going to talk about these two approaches. This was one project that I had in the lab with a master's student in collaboration with the bioengineering department at UCSD in a group at the University of São Paulo, Ariel Silber. And this student of mine, she collected information about all the proteins that were expressed in the different stages of the parasite. So based on proteomics and transcriptomics, she tried to use computational modeling to find out all the metabolic network and pathways from the parasite. And from that, we try to identify, okay, what are the weak spots here? What are the choke points that we can modulate with biological molecules to try to kill the parasite in an efficient way? So one of these important molecules that were associated with generation of energy in the parasite is the malic enzyme. So we published this work, and it turns out that another group in Brazil from LNBio had that specific enzyme, and they had already some molecules that seem to inhibit the enzyme. So we just got this project funded from CAPES in Brazil, and I'm working with them. We're going to do chemical modifications and try to see if these molecules can really be a good candidate to treat Chagas. I'll be running experiments in my lab to confirm that they kill the parasite in cells and in vivo as well. So about that second approach, we can grow the parasite in the lab. So we can generate this infectious form of the parasite that's called the metacyclic triple mastigote. And we use myoblasts, which is a cardiac cells, and we can do that infection in a well plate in the lab that is really the size of a cell phone. And this plate has up to 1,536 wells. And in each well, you can put a different compound. So in one plate that is the size of a cell phone, we can test 1,500 different molecules. So, that's a very good way to test things in high throughput scale. And that's what we're specialized. So we use robots to do all these addition of the molecules in the very tiny wells, dispense all the cells, the parasites. And then in the end, we take pictures of each one of the wells. So it's a very interesting way to do it, because in the end, what you have is microscopy images. Of course, we can't go back and see by eye what's happening to each one, because that would take forever, right? So we develop a software in the lab that can do the processing of all these images and translate these into numbers that can then be used to see, okay, which compounds kill the most parasites and do not cause any harm to the cells. So this is the principle that most of the projects that I have in the lab work around this platform. So I'll give you some examples of projects that we've done so far. This is a collaboration with Johnson & Johnson. We screened a library of 80,000 compounds. So a library is a set of small molecules. We call it a library. And this one was based on chemical diversity. And we identify, you see here, 1,351 molecules that had good activity, reducing more than 80% of the parasites. So the chemists will look at all these structures, try to cluster them when they have similarities and things like that. So we followed up with 2 series of compounds. And you see here the images. The big round objects are the nucleus of the whole cells, or the cardiac cells. And the tiny dots that you see around, these are the parasites infecting the cells. So when you treat it with benzinidazole, you see only the big round nuclei, meaning the whole cells are still there, but the parasites are clean. And this is one of these compounds from Johnson & Johnson. It also had a very good efficacy. So that's a starting point. We worked also with a research institution here in La Jolla, in San Diego, the Caliper at Scripps Research. They have a very interesting library called the Reframe. It's 12,000 compounds, and these include a lot of drugs that are already in use for different diseases. And the good thing about this is that if you find something that is active against the parasite, and if it's already an approved drug, you don't need to go through that phase 1 trial, because you know already it's a safe drug, so you can advance and test it in a phase 2 already in patients. So we identified a few candidates there. We have to now advance this and try to make sure if it works also in animal models. So this was published. We do also like to work with natural products. So this was a project with Oklahoma University. They have a library of fungi-derived natural products, and these are fungi that you find on your backyard. It's a citizen science project that they had, and we tested this library and identify a very interesting molecule called Leucinostatin B, which is very potent against the parasites, in fact, but it's also somewhat toxic. So we're trying to optimize this to get it more active against the parasite only. So this again was published. We take advantage of being close to the Scripps Institution of Oceanography here at UCSD as well. They're world leaders and experts in marine biology, and they have groups of chemists there that generate this library of marine-derived natural products, so about 100,000 prefractions and pure compounds. And we found one molecule called galenamide A that is, again, very potent against Trypanosoma cruzi. For this one, we actually even know the target, which is cruzaine, a very well-known and described target for the parasites. It's cysteine protease. I'll talk more about that. And this was published. Again, this is in that stage of trying to optimize the molecule and making sure it works in the animal model. So again, against the same target, cruzaine, we have a collaboration, long time collaboration with the Brazilian group at Federal University of Minas Gerais. Professor Rafaela Ferreira, she works on this target, and we have developed different chemical molecules that target this cruzaine. So um, a very fruitful collaboration with another Brazilian group. Uh, so as you can see a model, I work a lot with labs that provide the molecules, and I do all the biological tests. So this is a collaboration with Northeastern University. It's a very interesting one. We started working on a drug to treat cancer called lapatinib, and they did some modifications because it was also active against the cruzi. So we did some modifications, and we're optimizing these to the parasite. But the original molecule came from a cancer drug called lapatinib. So this was published as well. Okay, so what do we do next after we identify all these molecules? I mentioned about the animal model. So we did something very interesting. We get the parasite, and we genetically modify the parasite with a firefly luciferase gene. So the firefly luciferase gene is what makes the firefly glow in the dark. So we basically have a parasite that can glow in the dark, and this is for real. So what we do is we infect mice with these parasites that glow in the dark, and if you go to a very dark room, you can actually see light coming from the parasite. Well, we don't see it with our eyes, but we use an instrument that can do this. So it's a basic a black box with a very sensitive camera. And this is the kind of images that you would see. These are mice infected. So you see all this light signal coming from the mice. This is treated with placebo. And if you treat the mice with benzinidazole, it clears the infection. So this is how we do the proof of concept in the molecules. And some of the projects have advanced to that stage. So this is a collaboration with Derek Tin at Memorial Sloan Kettering Cancer Center. So we work on alkaloids, and two of these alkaloids have been tested in the animal model, and they reduce about 84% of the parasite. It's still not as efficacious as benzinidazole, but it's a good proof of concept that we're going on the right direction. This is another very interesting project. So you know ChatGPT, when you use machine learning and artificial intelligence to do things. So this was applied in a drug discovery project, and this collaborator, Sean Atkins, he works at Collaboration Pharmaceuticals. He did exactly that, a ChatGPT-like thing that would predict what compounds could be good against Chagas disease. So he interrogated the library of 7,569 compounds, and we selected top 99, and I did the test in my lab. We got 17 that were actually active, and we advanced 5 to this animal model. And one of the compounds that we identified was this one called pyranardine. So again, it had 85% parasite reduction, which is very good. And it turns out that pyranardine is actually a known drug, and it's used to treat uncomplicated malaria. It's approved in Europe in combination with artesunate. So we were very excited because this was the first time pyranardine was described as a possible anti-cruzi or anti-Chagas drug. So we contact the FDA. Sean Atkins actually got an orphan drug designation for the molecule. And they said, if you can show in a chronic model of Chagas disease that this is not non-inferior to benzinidazole, you can run a phase 2 clinical trial. So we got a group in Brazil that was all set for the clinical trial. This chronic model of Chagas disease takes a year to run. So now we have all the data, we're analyzing this, and if it does show some efficacy to prevent the cardiomyopathy, we'll then run a clinical trial in Brazil. So very exciting. And lastly, I think this is the very last one. I want to mention about this molecule K777. So this again targets that cruzaine, the target that I said that's very well studied. Cruzain was, I think, identified as a target by Jim McCarroll, a former mentor of mine and former dean of the Skaggs School of Pharmacy and Pharmaceutical Sciences. So he published this molecule K777 as an effective compound against cruzain in 1998, I think. So the same year Myra was diagnosed. And this has been for a long time a clinical candidate. So this, I think, is from 2013. The NIH was saying that this should advance to clinical stage, but they could never get the funds to advance the molecule to clinical stage. It turns out during the pandemic, this molecule K777 was also active to treat SARS-CoV-2. And there is money to treat SARS-CoV-2. So they were able to do clinical trial phase 1. This molecule was licensed to a company called Selva Therapeutics. They renamed it SLV213. It's a safe compound, so it passed phase 1, and they're now running phase 2 for SARS-CoV-2. The good thing is that now we know it's a safe drug. We may be able to advance this to see if it's infectious against the cruzi. So very interesting story there, and we needed a second disease with more financial interest to advance this for Chagas. So just to summarize, I think it's very clear for everyone that we need new and better drugs to treat Chagas disease. I think that can be a conclusion. There is only one compound in a clinical development pipeline, fexinidazole, and we expect to see the results in the coming weeks or months. There is still very few candidates entering preclinical phase, so there is this big gap. So I think this was already mentioned today, but we do really need more investment to advance these hits to lead to preclinical candidates and move on the pipeline. I just want to say that at the Skaggs School of Pharmacy and Pharmaceutical Sciences, we have this Center for Discovery and Innovation in Parasitic Diseases. So in addition to Chagas disease, we have, I think, 8 different other diseases that we work with, parasitic diseases and 2 viral diseases. So it's really a great center to have all these in a same physical location. So it's based a lot on collaboration with other groups all around the world, but it's really a unique feature that we have here in San Diego. And this work that I presented today, as you saw, it's part of collaboration with many different groups contributing to this. So that's it for me. Thank you very much for your attention.
Questions for all of the speakers. I have one for you though, really quickly.
Oh, sure.
It just came in and it's above my pay grade. If you can read that. It's
Do you want to read the question and I answer?
Do you think that any autophagy modulators show promise for treatment? Do you know what that means?
Autophagy modulators.
Autophagy, there we go. See, it's above my knowledge level.
Yeah, so autophagy modulators, I think the question is probably about asking if there is anything we can induce autophagy on the parasite, so making the parasite commit suicide or something along those lines. It could be. I mean, the way we run the phenotypic assay, which is the target agnostic approach, if there are small molecules that would induce this autophagy in the parasite, we would definitely identify those as candidates. So some of the compounds, let's say the alkaloids that we identify, we don't know yet what's the mechanism of action. We know they're good at killing parasite, but we don't know what's the target. It could be a mechanism like this. So we didn't have time, but there's a lot of ways you can try to narrow down and investigate what is the target and mechanism of action of compounds you find through that method. But I don't recall on the top of my head if there is any specific assay that would look for autophagy modulators, if that answers the question.
The person on Zoom that asked it, thank you. Thank you for the great questions.